We are mainly involved in development of wheat varieties for most of the agroclimatic zones where wheat is being grown. And within that, I am taking care of developing varieties through conventional and molecular breeding aspects where we introduce resistant genes into our breeding program, and also have a diversity in terms of rust resistance in wheat is concerned. So in overall, we will be talking some advanced phenotyping strategies, what we follow to screen our wheat materials for APR, that is adult plant resistance, and also for seedling resistance, where this kind of resistance is governed by a major gene or the oligo genes are the major single genes. So if you look at the wheat scenario in today's world, we all know that it is a major staple crop of the globe, and it is about 1/5 of total calorific and protein requirement is borne by this crop, and more than 80 million hectares of land is under this major crop. And within India, we have more than 34 million hectares being taken up every year under wheat. And also it contributes to 19% of total cereal production. As we expect our future generations, we have to deal with changing climate and changing biotic and abiotic stresses is a major concern to us. And we have to also deal with increasing population, which will be nearly 9 billion by 2050. So we need to increase our food production and especially wheat production by 60%. And at the same time, as I said, we have major challenges like biotic and abiotic stresses, especially the diseases in wheat. They are contributing to up to 10 to 13% of annual reduction in production. So giving us a major target to breed for this particular diseases. So it is very fascinating to see the evolution of wheat in parallel to the evolution of humans. If you look at number of years ago, it has happened like 2 lakh years ago when modern humans evolved in Africa. And then just 12,500 years ago, first evidence of agriculture was seen on this earth. And in comparison to the human evolution, if you look at the evolution of wheat, it is very ancient. It is around 5 lakh years ago that tetraploid ancestor of today's durum wheat, what we call the wheat which we use for rava making and pasta making. So that is the durum wheat, a tetraploid wheat which originated 5 lakh years ago. And then it is about 8,000 years ago, hexaploid ancestor of today's bread wheat, that is Triticum aestivum, that is the hexaploid wheat, and we also commonly call it as a bread wheat, which originated. So evolution of wheat is very strong, and there are so many years of process of recombination is involved. And there are very much challenges of phenotyping and genotyping, especially phenotyping for any trait becomes very complex whenever the trait is governed by multiple genes and it is under polygenic control. And you all know that wheat is having 3 different genomes, A, B, and D genomes, so making the genome even more complex. If you look at the way comparison is there with other crops, the size of wheat genome is very big. That we call it as a monstrously big genome. It is about 17 GB. And if you compare that to humans, it is around 3 GB, and cow, if you take, it is around 2.7 or 2.6, and mouse it is 2.7. Even though it is a small animal, it is having a bigger genome. And corn, it almost comes to 2.2 or 2, and chicken it is 1.6 to 1.5 to 1.6, and rice it is 450 MB. So when we compare the wheat genome to the rice genome, there is almost a 35 times difference in terms of the genome size. So this again will explain what is the complexity we have with this particular crop. And almost 17 GB out of that, that 16 to 17 billion DNA letters what we have in this genome, nearly 80% is the junk DNA. That means that it is not coding for any particular gene. And when we talk about number of genes which are there in these different genomes like human and the crops, major crops like rice and wheat, it is around 50 to 1,50,000 genes. So in wheat, it is said to be having 1,50,000 genes, and humans and rice will be having around 50,000 genes, which are required for most of the growth and establishment of their crops, specifically in case of rice and wheat. So still the wheat genome sequencing is it is incomplete, and still we are left with some chromosomes to be sequenced. So there is a challenge that we have to go for a good phenotyping. Before that, we have to have a good genotyping platform. So for that, you all know that the old system of Sanger sequencing has been replaced by next generation sequencing platforms. And there is a methodologies to reduce the complexity of genome, especially for a crop like wheat, which is having a bigger genome size. So the techniques called reduced representation libraries and genotyping by sequencing, which are being used so as to reduce the complexity. And also, if you look at the way the marker system has advanced and high throughput, it has become, we started from very coarse map of our wheat genome. Then there are from using SSR markers. Then there are other marker systems which are added in Songus in 2008, and also now we are in the era of SNP marker system, single base pair marker systems, where we have 9,000 marker system or 90,000 marker system or 35K, which is readily available even in India to deal with this complexity. So especially whenever we talk about use of molecular tools in breeding, we always talk about how big your genome is, and also you also have a methodologies or the protocols to see that the genome complexity is reduced, so as to have a good correlation with your molecular information, and also you have to have a phenotypic correlation with the molecular data you're going to get. So as far as biotic stresses in wheat is concerned, there are so many diseases which are there. For example, fungal diseases like rust and powdery mildew. I'm not going in detail into these, but today we are dealing with rust. You all know there are three major kinds of rust: stem rust, stripe rust, and leaf rust in wheat. And all of these have a specific coding patterns, and they are very biotrophic. They need a living tissue to survive, and they are widely adapted. And they have the ability to reproduce, and also they have the ability to evolve into new races. That means that there is a chance for a genetic recombination, so that new races are created, and the breeding has to be very much continuous to identify new resistant genes and introduce into wheat varieties. So this is leaf rust. They have very different parts they are going to affect. Leaf rust normally seen on the leaf, and very rarely it will affect head, and it is very not only rare, it is not visible on the head and stem of the plant, and it will come up very well when the temperatures are 35 degree, and it is having ability to cross 42, cause a loss of 40 to 70% yield losses whenever it is in very high disease pressure in the field. And the next is yellow rust. It is a cool loving rust, which needs 90 to 30 degree for its better establishment, and it has the ability to cause 10 to 70% whenever there is a lack of management practices. Either you use chemical or use resistant materials in the field. We have stem rust resistance, which normally visible on the stems of wheat, and it is more effective whenever the temperatures go below 20, go more than 25 to 30, and with more humidity, and created by incessant rains, and it is having ability to cause 50 to 70% yield losses. So they have their own effective way of controlling they're causing the disease. And just I want to show you, there is a 2-minute video just to understand this pathogen, if at all audible to everyone. So how this is affecting the crop in wheat.
Are under constant threat from invaders. This wheat crop stem is being attacked by a parasite. The parasite is called rust, although it is actually a type of fungus. The invading rust fungus has penetrated deep inside the plant tissue. As the invasion progresses, the fungus produces spores. These spores erupt from the surface of the plant. The spores spread, creating pustules, new sites of infection.
So the video is playing from the audio.
Soon the plant's stem and leaves are covered in fungal growth.
Thank you, sir. It is audible now.
Carried by wind, the fungal spores can travel great distances, eventually reaching other potential hosts. This spore must now find the nutrients it requires to grow. Sending out the germ tube, it seeks an entry point into the plant. A newly formed penetration tube breaks inside the stem, and the fungus extends further into the plant. Once inside the stem, another structure called the haustorium is used to penetrate inside one of the plant's cells. The fungus can now take nutrients from within the plant. It also begins to secrete small protein molecules called effectors. This is a critical time for the plant. If it can detect the fungal effector proteins, it can try to stop the invasion. The plant has specialized resistance proteins which act like an immune system. The resistance proteins can bind to the fungal effector protein. This binding event alerts the plant that an infection is taking place. Now that the parasite is detected, the infected cells are sacrificed, cutting off the energy supply to the invader. The fungus will eventually starve, and the plant will continue to grow. New strains of fungi are constantly developing through evolution. This can make some plants vulnerable to infection. Diseases like rust fungus have plagued crop production since people first began farming. Globally, infections of rust destroy 15 million tons of wheat each year. Because of this, wheat breeders must vigorously seek new sources of resistance to protect crops. Perhaps by investigating the interactions between plants and their invaders, we could one day prevent the devastation caused by rust fungi.
Okay. Yeah, that video was showing how these rust diseases are affecting the plant system, and they're creating a devastating losses. And when we have such kind of diseases, when there is a chances for evolution, faster evolution in the natural system, we need to go for development of rust-resistant genes. So that has to be very continuous, and every breeder will look after resistant genes which are there in the germplasm and diverse germplasm, and they screen and they see that how far they are going to be effective in coming years to introduce them into new varieties which are being released in this modern era. So there are two systems of rust resistant genes which are available in the natural system. One is all stage resistance, And the second one is adult plant resistance. Whenever we talk about all stage resistance, that is starting from seedling to adult plant stage, and also until the maturity, the plant will be free of any disease. And when we talk about adult plant resistance, though they may be susceptible during the seedling stage, but when they reach adult plant stage, then this kind of resistance is come to effective. So whenever you screen for all stage resistance, that we also call it as a seedling resistance, that it has to be done in seedling stage. And for seedling stage resistance, you have to take the most prevailing pathotypes which are available in the natural system. Then you have to have a controlled evaluation of disease response under controlled conditions by giving the temperature, required temperature for the specific pathogen. As I said, stem rust, leaf rust and yellow rust, they have stripe rust, they have very much different temperature and humidity requirements, and they take different number of days to express on the plant. So there are few important things whenever you go for seedling evaluation to check for resistant any major gene which is there for resistance against any three of these rust diseases. The first foremost thing is that you have to select the healthy seeds. Then you have to have a different sowing methods based on number of plants. You have a number of different genotypes you have to check. Either you can use the pots or you can use the trays to sow them so as to occupy more number of genotypes in a smaller area, because you will be growing under growth chambers or controlled environments. So growing condition, it also depends on the rust which you are going to screen, and the monitoring and evaluation is needed for any other environmental changes or the diseases are there other than rust pathogen. So plant preparation, as I said, that it has to be at a particular stage. You have to spray the inoculum, and there is a optimal inoculation time that even of like when the plant seedling reaches 5 to 10 centimeter, you have to spray the inoculum, and that normally reaches around 10 to 12 days after sowing. So pre-inoculation treatment just to remove the waxy cuticle on the plant so that the pathogen will penetrate very faster and very efficiently. And pathogen preparation, whenever we store the pathogen last year, a year back, and under the cold temperatures, that you have to take out and keep it at room temperature for some time before you spray them. So there are different methods of inoculation. One is lancet needle method, where you make a little damage on the leaf tissue so as to help the pathogen to fungus to enter into the plant system. Or you can use a toothpick method whenever you have a less amount of inoculum with you. Also you can use dusting method, just dusting the spores on the plants seedlings. So that can be done whenever you have a more amount of inoculum, and also you can spray the inoculum on the plants along with the water or any paraffinic oil. And brushing method is also followed whenever you have a good amount of inoculum with you. And incubation is very much important, that 95% of humidity is needed for most of the rust, and extended moisture condition even after 12 to 16 hours it is needed for the penetration of the fungus and entering into the plant system. So that pictorial representations actually are not given. And once the pathogen comes into effect and there is an infection, you can score your pathogenic reactions based on a scale which starts from 0 to the flake to 4. So there is a specific scale to measure the infection types, which starts from immune, followed by very resistant, resistant to moderately resistant, moderately resistant to moderately susceptible, then susceptible, and susceptible will be the last scale for the genotype which is showing completely 100% susceptible. When you have a heterogeneous infection, because whenever there are two pathogens which are causing the disease, then their reaction on the plant may be different. Such kind of in that situation, you may be giving XYZ next to them. So these are some of the examples of how we score. As I said, 0 is not having any flake or any chlorosis. As it was explained in the video, whenever the pathogen enters into the plant system, the plant receptors identify the pathogen, and it will trigger the response, that cell death response, whenever the plant is having a resistant gene within that. So that cell death response will going to create the chlorotic area on the leaf. That means that your plant is having a resistance. So whenever you get a good amount of pathogen on the plant, that means that there is no resistant gene present, and it is giving 3 plus to 4 is the reaction for whenever the pathogen is flourishing without any resistance from the plant system, and that indicates that there is no resistant gene. So this is also one more example for black rust, brown rust, and stripe rust. And you can see that there are some flakes without the pathogen, and at the end, whenever you go to 4, there is a good amount of pathogen on the leaf. That indicates that there is no gene here. And in this case, there is a gene which is present to show a resistance reaction. So adult plant resistance, as I said, it will be a response of the genes when the plant system, plant will achieve adult plant stage, that we will be now normally doing under the field conditions. So this is normally done under the field in the open environment condition. And the scale, modified Cobb scale, that is a specific scale with intervals, fixed intervals of 5 to 100% of infection to ensure uniformity of scoring across the globe, is adopted by CIMIT, which is there in Mexico. So there are some few things which has to be careful whenever you take up your field trial. One is site selection, and it has to be close to the rust hotspot. Either it is a stem rust or it is a leaf rust or the yellow rust. For normally yellow rust, we go to hilly areas in the off season like Leh Ladakh, and for leaf rust we go to Wellington, which is in southern part of India, and for stem rust also southern part of India and central part of India. It is normally seen for these two diseases, leaf and stem rust. Experimental design, it depends on the amount of land you have. Most of the time we go for augmented design to screen more number of germplasm for the disease reactions. And also inoculation can be done whenever the plant reaches the adult plant stage. And you need to have a spreader rose that for any disease, you must be knowing that wherever the pathogen is moving either through water or it is through air, you can use the spreader rows in the border so that the inoculum is multiplied to a maximum amount, and that will be spread onto the genotypes which you are going to test. So overall disease evaluation, as I said, we are adopting 50 to 70 Jadax growth stage, which starts from the tillering to the year-end emergence time. That time we go for disease severity estimation, and severity assessment is done. As I said already, modified Cobb scale is used 0 to 100%. It indicates how much of the leaf area it is covering, and the resistant categories are measured from very much resistant to susceptible. In between we have a moderately resistant. And there are some signatures on the plant. There are some signs of the plant which shows that there is some resistant genes present for rust resistance in wheat, that is leaf tip necrosis. Whenever adult plant resistance is there, for example, there are some genes, namely LR34 and LR67, they show leaf tip necrosis. Without screening for the disease, you can say that there is a resistant gene present within that. Yes, I was giving example for Zadok scale. As I said, it is 50 to 70. We start from a year emergence to milk development. So this is the exact stage where we measure the disease severity. And as we go forward and we go earlier, then the disease establishment will be not very uniform, and scoring will be much difficult in other stages. So for adult plant, as I said, we have highly resistant to highly susceptible, and in between moderately resistant, intermediate reaction, and moderately susceptible. And these reactions are very specific and unique to the resistant genes. There are some genes which may give you HR reaction. There are some genes which may give you moderately resistant reaction. There are some genes which may give you intermediate reactions, and there are some genes which may give you moderately susceptible. And apart from this, whenever you score it as a highly susceptible, then that indicates that there is no a single gene which is present in that particular genotype to show a presence of any resistance. This is a different scale. Will not go into detail. And if you explain, as I showed for seedling resistance and major gene resistance, this is for adult plant resistance. What we score for this is example of yellow rust. So if you score as a very resistant, the leaf has to be very much clean without any infection. And when you score for resistance, there is a yellow flecks or the cell death is present on the cell, that means that the pathogen has come, it has invaded into the plant system, but the resistance reaction has triggered a cell death mechanism so that the pathogen has failed to establish on the plant system. So this also gives a resistance. And also we have RMR, though the plant has resisting, but there is some spores which are present on the plant. That means that there is a gene which is not completely resistant to a particular race. It is showing a little bit of susceptibility. And in between, we have moderately resistant to moderately susceptible, and again moderately susceptible, then moderately susceptible to susceptible, then susceptible again, which is leaving some of the green areas where pathogen has not established. And then finally, we have a category called very much susceptible. You can see there is no much of green area present in this leaf to show that it is showing that plant is not at all resisting the pathogen to establish on its system. That means that there is no resistant gene which is present in this particular genotype. If you look at from very resistant to very susceptible, these are the different individual genotypes we have taken just to show the presence of different genes in these lines. So this is a CIMMYT developed phenotyping system, and whenever you measure how much of the leaf area the pathogen is covering. So if you look at here, these three stripes, it is showing 5% infection. Here it is 10% infection, as so on. And whenever the infection is very high, it is going to 100%. So it is also for stem rust, which is commonly seen on the stems. And again, we score from 5 to 100% based on how much of stem it is covering. And based on the reaction of resistance to moderately resistant, moderately susceptible to susceptible, we are going to screen our breeding materials. And again, this is for leaf rust. Whenever you see the rust in form of a brown spots, that is exactly a leaf rust. And there also we score our disease based on the amount of area it is covering from 5% to 100%, right? And the whole thing, whenever we measure the resistance as a R, we have a response value, calculated response value as a 0.2, and moderately resistance, we have a 0.4. By using these values, we calculate coefficient of infection, that to have a cumulative value from disease severity, as I said, 5% to 100%, and also to combine the infection type, like resistant to moderately susceptible to susceptible, these values. Whenever you score a particular genotype as a 10 MR, that shows that 10% of leaf area is covered by the pathogen, and MR is the moderately resistant. Even there is a pathogen, but plant is also resisting. MR is having a value of 0.4 response value. You multiply this disease severity value as a 10 into 0.4, and coefficient of infection is 4. Yes, the value goes towards 0, and yes, the value goes towards 10, that will vary your coefficient of infection values. It almost goes to, it will reach to 100, right? So towards, as it goes towards 100, it will be more of susceptible, and as it goes towards 0.2 and 0, that means that your plant is 100% susceptible. So these are some of the gene discovery work we have done in my postdoc time in Australia. So gene postulation is normally followed whenever you have any disease, for example, either it is in rice or any pulse, or if you take, or any oil seed you take, and you have a new pathogen which is emerging, like a powdery mildew has entered into a new crop wherever it was not there. Then what you will do that every gene which is showing resistance to powdery mildew will be having a different reaction. It will not be 100% giving, all the genes may not be giving the same reaction. So yes, it is given in the example LR1, LR2A, LR2A. These are the genotypes which are having these genes. So we have a differential set which is starting from LR1, leaf rust resistant 1 to leaf rust resistant. Now we have nearly 100 genes have been identified in across the globe. So the 100 genotypes which are having that particular gene will be giving a separate reaction, different reaction to a particular race. So that is study to study the presence of any gene in a new material. So that is a gene postulation. You all know that the population development which is required for mapping So we are normally using recombinant inbreed lines, backcross populations, double haploids, and F3 populations. And there are some advantages and disadvantages of these. So whenever you target for chromosomal location of the gene, bulk segregant analysis can be readily used, and advanced methodology is selective genotyping. Wherever you genotype your resistant and susceptible bulk and go for mapping. I'm not going in details into this. You all know that once you have a genotypic data, once you have a phenotypic data within your population, then you go for mapping. So either you are mapping a major gene or you are mapping a QTL. Whenever you are mapping a major gene, it is very, very simple. When you do that in a seedling stage, you will follow either bulk segregant analysis, or also you can use high throughput QTL-seq analysis, which is a replacement for bulk segregant analysis. And you can use the genotypic data to map the particular gene which is there in the population. And there are some softwares like Map Manager QTX or Map Disto also we can be used to map these genes. Even ICIM software can also be used. For mapping a QTL, you all know that that is measured whenever the plant is under adult plant stage, under field conditions. And here, accurate phenotyping is very much important. For every gene, the expression of that particular gene, considering the temperature, the light, and the stage of the plant, it will going to vary. So multi-year testing and multi-growth stage testing is very much important, and high throughput phenotypic data is very much important to map these QTLs and genes at a 100% accuracy. And for QTL mapping, you can use QTL cartographer or map maker or even ICIM, which is available freely. Those softwares you can use it. So one of the example which I want to give you before we went is that one gene we discovered during my postdoc, YR75, which is a yellow rust resistant gene. If you look at the reaction which is giving the susceptible genotype, Niabin 3 was giving 3+ reaction, and it is giving 11C reaction. That means that it is having chlorosis, and there is some amount of phenotype, the pathogen which is present, and there is a selective cell death on the plant leaf of the plant. So on a variety called X. So this gene we have mapped on chromosome 7A, and we have also developed a marker for this this gene. It is already cataloged as a YR75, and this work was published in Theoretical and Applied Genetics tag paper. And one more susceptible gene, here you can clearly see the resistance reaction of a plant, which is showing a chlorosis, very much chlorotic area. This is a leaf rust. That's why you can see the brown spots on the leaf. And wherever it is a resistant, it is having a chlorotic area, and also there is some amount of pathogen is also produced. So that means that the resistant gene is not completely resistant, but it is showing a moderately resistance reaction. And here in this plant, if you see, it is 100% infection on the plant leaf of the plant, and there is no chlorotic area. That means that plant is not resisting to the pathogen, and there is no cell death. That means that this is having no other gene. Then we crossed these two plants and developed a mapping population. This plant was giving 3+ reaction. That means that 100% susceptible, and this was giving IT infection type as a fleck. The fleck means the chlorotic area. 11 plus is the plus is where the spore is developed. So based on this phenotype, we develop a population and we map this gene. And if you look at the segregation, also it was showing that it is a major gene having a recessive gene action. And formally, this gene was named as LR82, and this paper was published in Gene. Why I am taking these two examples is that if you look at this variation, these populations were developed very, very early, but they were not tested under a particular environment. You see, whenever you screen for any disease, as I said, the pathogen needs a particular temperature, the pathogen needs a particular stage of the plant. Even though those real populations are handled by many other earlier scientists or any other student, it may not be true that the population may not be having a resistant gene. Only thing is that you have to provide exact temperature, exact amount, environment, what the pathogen is required to establish and to give the reaction. So this gene was tested many a times. After 10 to 15 times of effort, we identified to be expressed at a particular temperature on the plant. So again, this is the example. As I said, highly susceptible was giving 3,3 plus reaction, and this homozygous resistant was giving 1,2 double C. This is a resistant reaction what we were seeing on these plants. So we use bulk segregant analysis using 90K SNP data, 90K SNP bead array information to map this gene. And there is one more study that was made to identify a marker for UG99. This is the most prevalent and most effective stem rust race which is available in many parts of the globe, but it is not available in India. It is not yet reached India. So there was a gene SR26. It is again based on the proper phenotyping of the pathogen. We could identify a marker called Cas224 to be segregating with this gene. And if you look at the map also, which was located on 6A, and we could identify Cas224 and 225, which were linked to exactly linked to the particular gene called SR26 block. And as I said, this SR26 marker we identified, and that SR26 marker gene we wanted to introduce into many of the Indian breeding materials. So there were three varieties: PBW550 and DBW17 and HD2733. So in those materials, we introduced this SR26 by using SNP marker CASP224. And if you look at this picture, it shows that wherever you see these orange dots, it is homozygous for that particular resistant gene, and these are some of the heterozygote plant which are in the green color, and wherever you see the blue color, they are negative for this particular SNP marker. So we have used CASP technology, competitive assay polymorphism, to screen this material. So I would like to thank you all for patient listening, and I also would like to thank all